Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of the show, bringing another really awesome guest for you today. On the show today, we have the honor of being joined by Dr. Karsten Eastman, who is the Chief Executive Officer and co-founder of a company called Sethera Therapeutics. They are a company that's focused on revolutionizing peptide-based drug development with this really cutting-edge enzymatic cross-linking technology. And their platform ultimately enables the synthesis of these really highly stable macrocyclic peptide molecules, ultimately focused on engaging targets that previously were considered undruggable. Trained originally as a chemist at the University of Utah, where he earned his Ph.D. Dr. Eastman has built a career at this really interesting intersection of peptide synthesis, of protein engineering, and so-called radical enzymology, which we'll be getting into. And his work focuses on understanding not just how enzymes choreograph these complex molecular transformations, but ultimately harnessing those principles to build these really interesting programmable drug-like molecules. Recently, in collaboration with researchers from University of Utah, Dr. Eastman and his team published in the Proceedings of the National Academy of Sciences this breakthrough discovery known as PapB, really interesting enzyme capable of installing these really sort of interesting staples into peptides in an enzymatic step to create these molecules. And this work opens up a really interesting, unique chemical space, which we'll hear a lot about in terms of the macrocyclic peptides. At the company, he's really focused on translating this into these next generation products and collaborating with other organizations to bring these tools to market. And honored to have him with us. So a lot to get into today. Excited to talk enzymes and all things peptide biochemistry. Dr. Karsten Eastman, welcome to the show.
Thank you so much. That was a very thorough introduction, and I really appreciate it. You've definitely done your homework, and I really am happy to hear all of that. So thank you for having me.
Yeah, well, I'm happy to hear that you've done it all, and I know it's while we chatted a little bit about enzymes on past shows, I'm really looking forward to doing a deep dive with you because what you're doing is it's really fascinating. And I'd like just to really just start off at the beginning. I think our audience knows a little bit of what enzyme is, but I'd like to hear more about sort of the point where you sort of fell in love with enzymes. Obviously, you are a believer that these are very important tools for sculpting matter. Was it the speed, the efficacy, the intricate molecular machinery that enzymes represent that got you so fascinated in the first place?
Well, enzymes are nature's molecular machines. That's what they are. That's what they do. And the thing that made me fall in love with them was the fact that they manipulate physical reality with types of precision that in some ways is borderline impossible to match with traditional organic chemistry. Enzymes enable types of reactions with control, even in very complex environments that otherwise you can't access. And that idea that there's this type of machine that's so small but so precise is really what made me fall in love with enzymes and enzymology.
And as we sit here in 2026, and obviously we'll be drilling down into healthcare and pharmaceuticals and all that, but the enzyme market is pretty big. I mean, it's
It is.
tens
It's
of
huge.
billions of dollars. It's projected to be growing quite a bit. a lot of it isn't, I mean, obviously we know about it in healthcare, but a lot of it is other things. I mean, we just did a show actually with the folks over at Elanco, the animal health company, developing really interesting enzymes to help cows digest more efficiently so
Mhm.
they don't produce too much CO2. Obviously, the whole area of detergents and food and beverage products in terms of fermentation. You talk about the enzyme market in general and how you
Sure.
look at it. Clearly, you want to have the blockbusters in pharma, but talk about the space in general for the audience.
Yeah, so the global enzyme market, as you mentioned, is massive. It goes everywhere from industrial biotech, you have it in diagnostics, therapeutics, synthetic biology, and as you mentioned, it's big in agriculture as well. But the way that we're really focused on it is in more the therapeutic space, like you mentioned. And here, it's enzymes can actually be used directly. So there are enzyme replacement therapies. So say if a person has a particular hereditary disorder where they're not producing enough or the right form of a certain enzyme, and that can lead to some metabolic disorders, you can actually use enzymes directly as the therapeutic, which is a great type of therapy. Extremely expensive and very difficult to do, but it is transformative in what it can do to make their lives better. They're also used indirectly when you're thinking about, say, biomanufacturing or antibody engineering. Specifically, for many small molecule therapeutics, there are enzymatic steps to enable the correct transformation of the precursor to the actual therapeutic. And then there's also being used somewhat, this is newer, as programmable tools. So the idea of, the CRISPR-Cas type editing, where you have different machinery that guides, but you also have enzymatic steps where you have to essentially cut something out and put something back in. And this goes into, like I said, gene editing, post-translational modifications, and even in some ways like targeted degradation. I also think a lot of growth in potentially for even more engineered and programmable enzymes. And this sort of starts to get into the area of AI and enzymes, which there's still a lot of work that needs to be done here. AI is really good at making binders of, so say, proteins that bind really well, but making something that can very precisely catalyze a transformation is an area that I see a lot of work in the near future being done on.
Mm-hmm. It's interesting because yeah, that leads into sort of the first part of where I want to go with talking about PapB. And
Mhm.
before we get into all that it does, and sorry, it's promiscuous and radical, and we'll talk about that in a little bit. But let's just talk a little bit about where this comes from, because the
Yeah.
word PapB, you found this in something known as a Pannabis
Well, yeah.
polymixa.
So so this enzyme was actually very first discovered by Douglas Mitchell when he was at UIUC. He was the one who actually published the first paper on it. And what I want to highlight is, in many cases, is the discovery of what nature does in its natural context. But then sort of expanding that natural context out to see what it can achieve outside of it is really where we took over. So in nature, as you mentioned, PatB comes from Pennibacillus polyxema, and it was found by doing some genome mining, and there's a lot of things called small open reading frames, which typically are unannotated, meaning we sort of discard them. We don't think of them as actually being a gene. But in this case, when you have peptides, because this enzyme works on peptides, you have to consider those small open reading frames. And so what originally happened was Doug Mitchell's lab found the enzyme. They've said that, okay, in nature, this is its substrate and this is what it does. And then we sort of took over the mechanistic studies. But when we took over the mechanistic studies, we started asking some other questions of, well, if this is what it does in its natural context, can it actually be expanded out and do other things? And I think that this is a really important thing for us to consider is enzymes typically, the way that the field has been for a long time, is one substrate in, one product out. And that makes sense. That's how enzymes have been evolved to work. You want this really exquisite selectivity for just its substrate, and then you just get the product. Having a promiscuous enzyme from a historical context is actually really rare. And that's sort of this, that's really how it got started. Is we saw something that had a use in nature, and then asked the question, hey, can we do more than what it does in its natural context? And that sort of started the journey of all of these really surprising results.
Hmm. And then just one step before we get to the results, in that process, just for interest of the audience, and you're
Hmm.
talking about clearly how the sort of the discovery process in enzymology has shifted, from I guess sort of broad screening of microbial diversity and so forth in the natural world 100 years back to more AI and the genomics and all that nowadays. Was it like how many enzymes did you have to look at, let's say, before you say, hey, PapB is doing something kind of unique versus Pap A through Z? Or was it millions of enzymes? I mean, typically, in the typical day in your lab, how many different enzymes are you studying before you pick, oh, wow, this one's really doing something neat that we want to further investigate?
Right. And there, I think that that's where a lot of this genome mining comes into play. So one piece that I view, and this is just an example of, so let's say you have some sort of repeating motif, and I think PatB is the really good example. PatB has this spacing of where you have a cysteine residue, and then you have a acidic residue, so an aspartate or a glutamate. And then there are multiple cysteines and multiple aspartates and glutamates. So in its natural product, it actually forms 6 different thioether linkages. So there's 6 sulfur to carbon bonds, and all of those take place at a really specific location on the peptide. Now, if you're doing 6 different transformative events in the same peptide, you can sort of get an idea, okay, maybe there's some flexibility here, or at least that's how I thought about it. So when you're looking at the genome mining context, one thing that I think is important is, do you see a pattern that could perhaps be exploited? And that's where there's a lot of your own thought process and your own understanding of what transformations might be happening. There's a lot of different enzymes that have been studied. There's still hundreds of thousands, at least in the radical SAM, that's where PatB, that's the family it belongs to, superfamily, that have yet to be explored. So really, to be blunt, with PatB, we got extremely lucky in that this was an enzyme that was as promiscuous as it was. Really, when I was working with Vahe Bandarian, who was my graduate mentor, I did my PhD under him, when I first suggested some of the early experiments, he basically told me, that's the stupidest thing I've ever heard. It's not going to work.
Yeah. What are you gonna do?
Yep.
Oh. But no, okay, so this obviously this segues perfectly into your dissertation, and let me just ramble this one off. Substrate scope and mechanistic studies of thioester forming radical SAM ribosomally synthesized and post-translationally modified peptide maturation.
Yep.
Um,
It's a mouthful, isn't it?
it's a mouthful. It leads to the formation of Sethera, but ultimately, summing it all up, you find out really cool things about this promiscuous radical enzyme. You used the term, I
Yep.
think, at one point, promiscuity with control. Very
Yep.
important when we want to create a tool out of this. Take us into the dissertation and some of the
Sure.
aha
Well,
moments.
yeah, well, let me first back up and let's tell the audience a little bit what each of those segments of the
Please.
words mean. So radical SAM, that was S-adenosyl-methionine. I'm sure that some people have heard of SAMe as a supplement. I mean, people use it and take it all the time. So radical SAM maturases or radical SAM enzymes, they use SAM and a radical mechanism to do lots of different things. That's the easiest way to think of it. It uses SAM as a cofactor, and then it's uses that as a piece of the radical initiation to do all of these complex transformations. Then you mentioned RIPS, which are ribosomally synthesized and post-translationally modified peptides. All that means is you get a peptide off the ribosome, you have something else that changes it. So we really combine those two things. You get a peptide off the ribosome, and then it's a radical SAM enzyme that changes it. So, and in this case, we're forming a thioether. So that's really the fancy way of looking at everything that was in my dissertation. And really, we looked at 3 main things. The first is substrate scope. So we sort of said, okay, PatB does this in nature. How far can we push it? right?
Mhm.
That's can we do all of these transformations, say make the macrocycle smaller, make it bigger, see how far we can go in making it bigger. Are there any residues that don't work? Or can we put in unnatural amino acids? And is all of that okay? And the blunt short end of it is yes, all of it works. So we found very, very few stopping points, and that's what surprised us. And then the second piece of this is, the whole mechanistic insight. When a lot of people are in a chemistry PhD, depending on their field, they're studying the mechanism by which something occurs. And I think for a lot of people who aren't really deeply ingrained into the sciences, they think, okay, why are you studying this? Why should we care? This is so niche, etc. But when you study mechanism, you can gain a lot of insight into how, say, when you see the actual chemical transformation and you gain insight into it, you learn how to manipulate it. Right. And so even though mechanistic studies by themselves to the, layperson seem very boring, there's a lot that can actually come from mechanistic studies. And that's what we were able to see. And then finally, we demonstrated that the enzyme could replace more labile bonds, so say a disulfide, with the stronger thioether in an already on the market therapeutic. So that's really how it started, and those are really the three main areas that my dissertation looked at. But it did directly lead towards the formation of Sethera, because we realized that if, hey, we can take this disulfide that usually is linearized in cells and replace it with a thioether, which is much stronger, there is a great value proposition there in terms of being able to modify or manipulate already on the market therapeutics. So that was where our thoughts first started.
And Chris, can you say a couple more words about that? Just because I want before we get to macrocyclic peptides, which
Sure.
is the core focus now. Along the way, and just reading about some of the steps. I mean, you first used it in sort of these smaller non-peptide fragments, and you moved on to an interesting coumarin derivative, and then you played around with an FDA that you mentioned FDA approved drug, this setmelanotide, which is a sort of a rare disease or a genetic obesity disease. But again, can you just take, I mean, for sure, the lay audience that's listening, because I think it's kind of cool how,
Yeah.
okay, we start with these basic compounds and we chose a natural product and we did a protein. It all lines
Sure.
up. I just wanted to explain that part as
Yeah.
well, because I think it's kind of
Sure.
interesting.
So we really did the majority of the work on peptide-based therapeutics, since the natural product was a peptide. What we did is we essentially took this very, very long form of it that was 45 residues in length, and then only actually looked at what we called minimal substrate PapA. So PapB matures PapA. That's how it worked. And that's where we started. And the main reason was studying a single bond formation event is a lot easier than studying six, like is in the natural product. And when we started looking at one, that's where we probed it with a lot of different questions. The questions were things like, can we make the size of the macrocycle smaller? Yes. Can we make the size of the macrocycle bigger? Yes. Can we stick in unnatural amino acids at every single location in the context of the macrocycle? The answer was yes. And then we started looking at, okay, we're sort of limited here because we're going from a cysteine to an aspartate or a glutamate. Can we, say, go to a different residue that is, say, a C-terminal glycine? A C-terminal glycine looks very much like the side chain of an aspartate, and we saw that there is cyclization events that occurred there. Now, can we stick in unnatural amino acids at the C-terminal and still have it be processed? The answer there is also yes. So we systematically probed every single way that the enzyme could possibly result in a transformative action. And really, there were very few places where it didn't work. One of the really unusual things, so for any of the peptide or protein chemists, leucine, isoleucine, and valine have some similarities in their structure. They are all very branched and bulky. But when we considered the amino acids at the C terminus, Valine did something weird. It went to the beta carbon instead of the alpha. Isoleucine didn't work at all, and leucine went to the alpha carbon. So that's just some of the more technical pieces of we saw some unusual things happening, but we sort of laid out all of these rules. And now that we understand the rules, we started on seeing how it could be used in commercial applications.
Let's now setting all that up, let's now reintroduce the audience to briefly to the principle of undruggability, because
Sure.
this is very important for where your products come in terms of the peptides. But
Yep.
again, we have a very large segment. I mean, we discovered a lot of stuff from the Human Genome Project and the proteome and all that.
Yep.
Unfortunately, a lot of it not easy to dock small molecules into. Take us into what undruggable means.
Sure. The easiest way to think about it is size and surface area. Small molecules, by their very definition, are small, typically under 600 Daltons, and you do that for a variety of reasons. One of the main thoughts is how do you keep the small molecule passively permeable, orally bioavailable, and give it a nice PK PD profile. And there's a lot of rules, one of them being Lipinski's rule of five, which there have been lots of ways of breaking that as time has gone by. But really what it is if this is your protein target, right? And you need to say stop it from interacting with this other protein target, a small molecule, which could say be like the size of my pinky, is not going to block that interaction. That's, I think, the easiest way to visualize it. Small molecules are small. The nice thing about peptides, and this is why we're moving into this whole undruggable space, if you have something that's small, you can't block certain interactions. If you have something that's bigger and also mimics certain types of those same interactions, suddenly you can start to drug these undruggable targets. And that's what peptides can do. Since peptides are small chains of amino acids, they retain many of or similar properties to proteins. Proteins are just large chains of amino acids. So we can exploit the fact that, number one, the peptides are bigger than small molecules. And then number two, they interact with proteins very similarly to how proteins interact with other proteins. So there's lots of nice properties that can be exploited to make historically undruggable targets druggable.
And this is, the beauty of where your work comes in because you're focusing on macrocyclic peptides. So, you're bridging the gap between the small molecule and the large biologic. And you were saying all this unique dynamics about specificity and affinity and so forth. Again, you have a very interesting platform there. And I know, Kurt, I guess you're working with different companies and so forth, but it really, you talk about the polymacrocyclic peptide discovery platform. It's got a library, it's got AI, it's got all sorts of other tools attached to it. Take a little time to sort of walk through the architecture of this and what Sthera is basically based on in terms of its
Sure.
platform technology.
Sure. So when we started Sethera, we had some initial thoughts, and those thoughts very quickly changed, shifted, and pivoted. What I did after we formed is I went to the Bio conference and to several other conferences to try to figure out what the pain points are or what are some of the concerns in the peptide space. There are quite a few peptide platform companies that are currently out there. One of the focuses of essentially all of those peptide platforms are macrocycles, but essentially all of those companies are limited to a single macrocycle. And that's because controlling chemistry outside of a single macrocycle is a very difficult thing to do. So for, again, just a visual, here's your macrocycle. You can have all of this fantastic chemical diversity on the outside, but biology 101 is structure equals function. And if the only baseline architecture you have is a macrocycle, that inherently limits what you can do. And the next example of that is, if this is your complex protein surface, you have a macrocycle, you're really only able to engage with one segment or portion. You can't necessarily interact with multiple portions at the same time. This is really where Sethera comes in because we can generate higher order architectures, meaning This is where the polymacrocyclic piece comes in. We can generate these absolutely massive numbers of barcoded peptides that have all been enzymatically processed to then have this higher order architecture and then run them over targets. This means that we can start to access antibody-like binding, so multivalent binding, with peptide-like size. So that's really where the platform and the focus has been, is we are building as diverse architectural libraries as we possibly can. And some of what's falling out of that is, I guess, surprising because it hasn't been seen before, but from a biology perspective, isn't too surprising because, again, as I said, structure equals function. We're not only seeing amino acid enrichment that we're pulling out of our screens on certain targets, but also target by target architectural enrichment. So rather than certain targets being more amenable to, say, all architectures evenly, we can see enrichment of, say, architecture 2 and architecture 5. And I think that's where a lot of the power in the platform comes from. We're not just exploiting diversity of amino acids, we're exploiting diversity of architecture.
And I see, you're out there, you're learning a lot about what the industry needs. Could you say something about, I mean, I saw, I think your most recent paper, you're doing some work with GLP-1, which is
Yep,
obviously a hot theme nowadays and an
it
interesting
is.
target to play around with. But do you come up, I mean, do you come up with some of the interesting targets in-house yourself? Are you only sort of working with sort of the discovery organizations of pharma companies now? Talk a little bit about, do you think beyond pharma? Obviously, there is lower hanging fruit still out there in terms of all the other parts of the enzyme industry that we've spoken about. You might want to be staying away from that or not, but I don't know. But take us a little into the business model.
Sure.
What we are
Right
thinking.
now, the business model is we want to be the preferred screening and initial development partner for pharma companies. I think that our screening technology, we've demonstrated that it's working and it's working really well. While internally we do have thoughts of going after our own targets in, say, uh, rare cancers, orphan indications. We don't want to be seen as a competitor to big pharma because we really don't view ourselves that way. We don't want to explicitly get into all of the weeds of drug development on our own. But we recognize that the further we take individual Sethera assets, so say we move something towards phase 1 or phase 2 in one of these rare cancers or orphan indications, it gives more validation to the platform as a whole. So that is one of the areas that we're thinking about and focusing on, is both being the screening partner, but also showing that our peptides and our molecules themselves can, say, go towards the clinic and be very safe and effective.
And when it comes sort of to obviously the PapB discovery is amazing, and you're going to be doing great things with that in terms of the platform. What else sort of comes? I mean, do you have other internal enzymology discovery initiatives? Is what I'm thinking, because I don't know if you saw, we did an episode with Craig Venter about a year and a half ago, and he's out there, taking his boat around the world and scooping up all sorts of microbes all over the oceans. And then, wow, we got the billions of potential new enzymes. So how much of your energy are you now devoting beyond PapB to saying, hey, what are the other interesting PapB type stuff that's out there? Because clearly, I mean, I don't think you Sethera is going to be based on just this one radical
Yes.
mechanism. Maybe
So,
take us
so
on
I
that.
mean, yeah, right now, I think that people probably think of Sethera more as a peptide company, but I think that that's not necessarily accurate. I think we truly are an enzyme company. I think that we've just had some serendipity in terms of which enzymes we've worked with first. But I recognize that there's a lot of transformative enzymatic space that has a lot of value. So what I see in the future as we continue to develop the screening platform is also starting to develop enzymes for, say, the aging space. I think that that is one of the areas that interests me the most, is in terms of repair to the body. Enzymes are the best and most fantastic tool to enable that. is a lot of work in that space to be done. And I recognize that enzymes are going to be the tool to achieve it. So I still think that that's a bit of a far off goal in the future. We still want to ensure that we build this and further this very robust screening platform, demonstrate that these peptides have even more potential than we think that they have. But then in the future, I recognize that Sethera will remain in the enzyme space. And one of the areas that I think is most interesting will be in the aging area.
You know, we discussed a bit of that on the show in terms of some of the weird sort of extracellular junk that accumulates in our bodies that maybe we don't have the perfect enzymatic machinery to break down like other species do. And that, yeah, I think that fits beautifully actually into what you're doing, especially in thinking about not just peptide moieties, but oral bioavailability and, you know, sort of the, you know, what the industry is excited about. That just got me thinking of one other thing, actually, as we're sitting here. Anything
Sure.
interesting in your shop with regard to extremophiles and enzymes? Because I know that's always been a pretty hot or cold area,
Yes.
depending on where you find the extremophiles. But anything cool happening on that front, or is that sort of a more of a specialty enzymology niche?
Well, so there are employees at Sethera who have, a very special place in their heart for tardigrades, right? I mean, you
Or
can send those critters into space and pull them back, and they can be exposed to the vacuum of space, all of this radiation and everything else, pull them back, and they're still perfectly fine and perfectly healthy. And from a biology standpoint, really the only thing that can enable that type of robustness and survivability would be in the protein and enzyme contexts. So there are critters, and I mean that have a lot of potential to explore. And again, it's going to be in the protein and enzyme space. So understanding what is it about their composition that enables them to survive such extreme conditions. And the things that enable that composition are proteins and enzymes. So yeah, you're absolutely right. That is a very interesting space for enzymology.
yeah, and it's an exciting set of topics, and
Yeah.
you know, it's going to be again exciting to see how your platform develops. You mentioned, you know, you just a little while ago you were out at Bio, you know, talking to pharma companies and biotech shops and everything about collaborations. I saw that I think coming up in a little bit is the sort of the Peptide Drug Summit in 2020. I'm trying to see where that's going to be held,
That's
but
in
I know you're going to
Boston.
be. That's in
Yep.
Boston. Okay, you're going to be
Yep.
presenting at that. Where else are we going to be able to find you, Karsten, over the
So
next several months, of
we
2026?
have been really focused on getting the internal science developing in a few different areas. There's going to be some interesting news in the near future. Right now, the main spot to be able to catch me will be Bio in June. So I will
Okay.
be there. I'm not doing too many public presentations right now because we're trying to get a few things rolling with patents and a few things rolling otherwise. But probably in the fall is when I'll be back to making more public appearances.
Again, it's I love hearing stories of young companies like this, especially that come off PhDs and you know have the ability to disrupt the pharmaceutical industry in a major way. So it's very exciting what you're putting together there. I hope to be able to do a follow up with you later in the year as we hear
Excellent.
more about these initiatives. But really impressive work. I have to say. Again,
Thank you.
for everybody out there that is going to be listening to this episode across the various podcast networks or watching on the YouTube channel. Again, you've been spending time with Dr. Karsten Eastman, Chief Executive Officer, co-founder of Sthera Therapeutics, doing really amazing work at this intersection of peptide synthesis, protein engineering, radical enzymology. Karsten, again, I want to thank you for taking the time out of your schedule to come talk to us for a little while today and educate us on some
Of
topics
course.
that we haven't gotten into. Thanks for everything you do there for potential human health. And as we like to say here on our show, thanks for helping to create a better tomorrow through the technology developing. It's really a great story. And I look forward to continue to follow it.
Thank you so much. I really appreciate it being here.
It's great having you.